Learning outcomes. By the end of the lesson, students will be able to define nucleosides and nucleotides and distinguish between these two fundamental biochemical molecules. Identify and describe the key components of nucleosides and nucleotides, sugar, nitrogenous base, and phosphate group, and recognize the role of phosphodiester linkages in forming nucleotide chains. Illustrate the structural and functional differences between DNA and RNA, with emphasis on their nucleotide composition and biological significance.
Hi everyone! Welcome to this short but essential lesson on nucleosides and nucleotides, two of the most fundamental molecules in biochemistry and molecular diagnostics. Let's make this fun, visual, and practical, since especially you'll be encountering these terms a lot in labs and lectures. Let's start with a quick challenge. Look at these two molecules. Can you spot the difference? Here's a hint. One of them is more complete. The answer? The one with the phosphate group is called a nucleotide. The other is a nucleoside. So now, let's break these down even further. A nucleoside is made of just two parts, a sugar and a nitrogenous base, no phosphate. But a nucleotide has three parts, a sugar, a base, and a phosphate group. And that's the key that lets it form chains. Notice this. The phosphate group always attaches to the 5' carbon of the sugar in a nucleotide. This specific attachment point is not random. It's very important because it determines the directionality of the nucleotide strand. Later on, when we build DNA or RNA, the 5' to 3' orientation is what guides how nucleotides are connected and how genetic information is read and copied. So, the tiny 5' carbon plays a big role in how our genetic material is organized and functions. Now, let's talk about how these nucleotides connect. When nucleotides link together, they form phosphodiester bonds, which are strong chemical connections between the phosphate group of one nucleotide and the sugar of the next. These bonds act like the glue that holds the nucleotides together, forming the sugar-phosphate backbone of DNA and RNA strands. This backbone provides structure and stability, allowing the genetic code to be stored and passed on accurately. Is always sugar to phosphate, creating a stable, repeating structure. Let's zoom out a bit. Zoom in, zoom out a bit and see how nucleotides shape DNA and RNA. DNA uses deoxyribose and includes thymine. It's double-stranded and stores long-term genetic information. RNA uses ribose and swaps out uracil for thymine. It's single-stranded and works in protein synthesis like a short-term messenger. Time for a quick activity. Look at these three molecules and answer in your head or in your notebook. Which one lacks a phosphate group? Do you have an answer? Well, here it is. Nucleoside, since it has no phosphate. Since it only has a sugar and a nitrogenous base, it can't form the backbone of DNA or RNA. Which one uses uracil? Done thinking? Here's the answer. RNA nucleotide, since it has uracil. Uracil replaces thymine in RNA, which helps distinguish it from DNA during cellular processes. Which one comes from DNA? Okay, here's the answer. DNA nucleotide, since it has thymine and deoxyribose. The presence of thymine and deoxyribose identifies it as a DNA building block used for long-term genetic storage. Well done! These quick visual checks are great for active recall. Let's apply this to the real world. Imagine you're working in a lab. You're prepping a COVID-19 test and someone gives you a vial labeled nucleosides instead of nucleotides. Would that be a problem? What's missing? Yup, it's the phosphate group. Without it, you can't build DNA or RNA strands. RT-PCR tests work by copying genetic material, and they need nucleotides to do that. This is why understanding these molecules is not just academic. It's essential for diagnostics and healthcare. Now, it's your turn to show what you've learned. On any paper or your notebook, draw a nucleoside and a nucleotide. Label the sugar, base, and phosphate if it's there. Then, make a simple comparison diagram, like a Venn or table. What do they share? What's different? What's their role in DNA or RNA? This short task activates your memory and helps reinforce the key differences. To wrap it up, test your understanding through a short 5-question quiz on Quizzes. You'll get the link and QR code right here. It's fast, it's fun, and it'll tell you right away if you've mastered today's topic. You can pause the video and come back right after. Make sure to come back, huh? I'm watching you! Before we end, here's a quick recap.
A nucleoside is a molecule that contains a sugar, in this case 2-deoxyribose, and a nitrogenous base. A nucleotide contains a sugar and a base as well, but it also contains a phosphate group, which can form a bond with a sugar of another nucleotide. This is why nucleotides are the building blocks of nucleic acids, as you can see here on the example of DNA, where 4 nucleotides on each side are linked together by the phosphate groups. The alternating sugar and phosphate groups create what is known as the sugar-phosphate backbone, which is important for the molecule's stability.
A nucleoside is made of just a sugar and a nitrogenous base, while a nucleotide includes a phosphate group in addition. That phosphate is crucial. It's what allows nucleotides to link into chains and form the backbone of DNA and RNA. Without it, a nucleoside can build genetic material. Nucleosides serve as precursors, but nucleotides like ATP and DATP are the actual building blocks of life. So remember, if you forget the phosphate, you forget the function. What the function? What the function? Thanks for joining us today. Whether you're studying for a quiz, prepping for lab work, or just love science, you now know how to tell a nucleoside from a nucleotide and why it matters. See you in the next lesson. Bye-bye!